wanted to make a video to help you with the notes on mitosis. If you haven't already, stop and go back and watch some of those YouTube videos that I've inserted into the course. They are so good and helpful, and those people make videos for a living, and so they're much better than me. And so, I did want to make a video to kind of help you go over mitosis. And so it doesn't matter if you're looking at the student notes or if you're looking at the PowerPoint, that the words are the same and there's only a few pictures in the student notes. But for the presentation, for the video, I'm going to pull up the PowerPoint. Okay, so the cell cycle, this is how cells divide. And we're thinking about, skin cells, bone cells, all the cells that divide go through what we call mitosis, where they make identical cells. And that's what we're going to learn about. This is just a cool picture I put in there. Where one cell will start to divide and then just, divide more and then completely become two different cells. The cell cycle is an ordinarily set of stages that take place between the time a cell is formed and the time the cell divides. It's, the cell's life history. And so do me a favor, highlight, and I'll try to highlight as we go. The cell cycle is an ordinarily set of stages that take place between the time a cell is formed and the time it divides. And so when you're looking at this, you've probably learned this before. When you're looking at this image, you'll see that the majority of the time is spent in interphase, which is in between dividing. And then mitosis is what we call actually the division, and that's prophase, metaphase, anaphase, telophase, and then when it completely falls apart, we call that cytokinesis. So interphase is the time in between, when it's doing its usual functions. Most of the time is spent here. This is in between dividing. And so there's 3 stages in interphase. If you haven't already, write in like in between division. This is not the division. This is in between the division. There's G1. G stands for growth. S is where the DNA is replicated, and then there's G2 is like the next growth or gap. So G1 is the growth, the stage before the DNA is replicated. S is DNA, I think the S stands for synthesis, so the DNA is synthesized. And then G2, the cell synthesizes its proteins and it prepares for division by making its microtubules and things like that. And so it's like grow, DNA replication, grow. And then we talk about mitosis. The next stage is mitosis. The cell division state includes when it's actually dividing. It's called mitosis, right? And then the last step, when it completely falls apart, the cytoplasm is divided, we call that cytokinesis. The activities of the cell are controlled by its checkpoints or signals. And when we really get into this, it is so interesting to think about how your cell pauses and checks to make sure it's dividing correctly. Signals are used to ensure the steps of the cell cycle take place in the proper order. So checkpoints or signals are used. And then there's growth factors, or signals that stimulate the cell cycle to take place. And so, and you think about when you cut your skin, your skin's going to grow back and fix itself. You break a bone, it's going to grow and fix itself. Okay, so this is that same image but bigger. You've got interphase, which we said is 90% of the time, G1, S, and G2. And then there is, let's change it up, mitosis right here. And we're going to learn prophase, metaphase, anaphase, telophase, and then complete division is cytokinesis. Highlight for me that apoptosis is when the cell dies. When it's had all it can take, apoptosis is when the cell dies. Highlight that. The cell goes through a series of steps that bring about its own destruction. So apoptosis is programmed cell death, when the cell has reached the time where it's supposed to die. The cell breaks into pieces and is engulfed by your immune system, your white blood cells. Okay, when we talk about mitosis, that's the division, and cytokinesis, when a eukaryotic cell is not dividing, DNA is found in the chromosome. This is like a chromosome here, a doubled chromosome. When it's not dividing, it's found in its chromatin form. Let's see. Okay, so before is all. So your cell, like right here, this whole time right here, your DNA is in. Right here, this whole time, your DNA is in chromatin form. That's loosely coiled DNA, like just spaghetti and meatball looking DNA. When it gets ready, in prophase, it will begin to coil up tightly into the shape of a chromosome. That's what it's showing you right there. Before the mitosis takes place, in the S phase, the DNA is replicated. Now each chromosome has two identical double helix molecules, and we call these, so highlight this right here, sister chromatids. That's what these are, sister chromatids. So make sure you understand that. Your DNA is always in the shape of chromatin. That's loosely coiled DNA until it starts prophase, and that's when the DNA will tightly coil into the shape of a chromosome. And you can see it's got two sister chromatids. In the middle, it's called the centromere, and it's got that protein kind of hugging it on either side called the kinetochore. Here's the stages of mitosis. Memorize this. PMAT. Prophase, metaphase, anaphase, telophase. Here's an image of it. You can see in prophase right here, the chromatin becomes chromosomes. The chromosomes start to attach to spindle fibers right here in prophase right here. And then in metaphase, the chromosomes line up in the middle. Meta, middle, M. Anaphase, A, the chromosomes start to pull apart. See, if you analyze something, you pull it apart. A, pull apart. A, pull away. And then telophase is where the cell starts to divide. See, the actual cell is dividing. Cytokinesis is where it like pulls apart. And that's what I just told you. Prophase is where where do chromosomes appear? Chromatin condenses in prophase. Nucleolus disappears. Nuclear membrane disintegrates. Centrosomes migrate towards the poles. They start to form those spindle fibers, and then kinetochore fibers attach. So the chromosomes attach to the spindle fibers or the kinetochore fibers in prophase. Okay, so let's go over it real quick. In prophase, the chromatin becomes chromosomes. It says chromatin condenses into chromosomes. The nucleolus disappear. It just magically disappears, literally. The nuclear membrane disintegrates. That's some of the like the membrane around the nucleus disappears, and then the chromosomes start to move and they attach to spindle fibers. Okay, in metaphase, this is easy. The chromosomes line up in the middle. Chromosomes line up in the middle. In anaphase, highlight this, sister chromatids separate. Highlight that. You want to know, it's real important now that you understand that half of the chromosomes, the sister chromatids, separate. In the next chapter, we're going to learn about sexual cells dividing, and we're going to see that chromosomes separate. But right here, you're learning sister chromatids, so half of it separates. Anaphase is the shortest phase. And then telophase, you can see right here the cleavage. It starts to show right here. Spindle fibers begin to disappear. See, and this is like the opposite of prophase. In prophase, the spindle fibers appeared. Now the disappearing, mitosis is complete at the end of telophase. And so you can see this picture. That's prophase. Prophase. Nuclear membrane disappears. Still prophase, and metaphase, anaphase, and telophase is where it starts to come back. Okay, so let's watch that again real quick. So prophase, the chromatin becomes chromosomes, nucleolus and nuclear membrane disappear, spindle fibers form, chromosomes attach. Now that's metaphase in the middle. Anaphase, they pull apart. In telophase, the nuclear membrane reappears, nucleolus reappears, and then cytokinesis is where it finally breaks apart. Cytokinesis is the division of the cytoplasm. Highlight that. Okay, in the end, you have two identical, they call them daughter cells, but they're two diploid. The word diploid means it has all the chromosomes, 23 pairs. Diploid means 23 pairs. So you have two identical daughter cells or diploid cells. We talked about that furrowing. Two identical cells. Okay, there's cytokinesis in animals. And this, we don't have a cell wall, so it doesn't look like it has a cell wall. In plants, there's a cell wall, so they form this highlight cell plate. Vesicles containing a cell membrane components produced by the Golgi move and fuse to form a cell plate. That's a cell plate, make sure you know, is only in plants. Cell plate is in plants. There was no, that's because of the cell wall. Oh, there it is in, okay. Functions of mitosis. It allows a single fertilized egg, sperm and egg together, that's a fertilized egg, to become a multicellular human, right? The diploid cells, of course, I think, but just in case, the egg is much larger than the sperm. And the sperm puts its DNA inside the egg, and that becomes one fertilized egg. Now that one fertilized egg reproduces and reproduces by mitosis and becomes a baby. It's a miracle. Okay, it's also necessary for a cut or a broken bone to mend. We talk about adult stem cells that retain the ability to divide. So stem cells are cells that are not yet specialized yet. And so they're not bone cells or brain cells or blood cells. They're, kind of like you haven't decided your major when you go to college. They're cells that haven't decided what they're going to be, but they have all the DNA that could be anything, potentially. There's all different kinds, but for this lecture right now, we'll just know that they haven't yet decided. Haven't yet decided what type of cell they're going to be. Adult cells that retain the ability to divide. Examples: red bone marrow stem cells divide to produce millions of cells that later become blood cells. There's therapeutic cloning and reproductive cloning. Therapeutic cloning uses stem cells to produce human tissues, and then reproductive cloning uses embryonic stem cells to produce new individuals. So really, really cool. Pause the video. I do have some more information about stem cells in another assignment, but stem cells are so cool and interesting. I'd say pause the video and read about those if you're interested in that. Okay, let's make sure you understand, talking about, this is a picture of reproductive and therapeutic cloning. You can see Dolly the sheep. I remember I was a little girl when Dolly was made. It was such a big deal. My mom was a science teacher, and so she taught me all about it and told me about it when it happened. But then here is therapeutic cloning that can make nerve cells, blood cells, muscle cells. Okay, we talk about chromosomes. Remember I told you chromatin is loosely coiled. It looks like spaghetti and meatballs. Chromosomes are tightly coiled in the shape of an X. Except that didn't go over well. And so you can see chromatin loosely coiled, chromosome tightly coiled, 140 million nucleotides. Nucleotides make up the chromosomes, and we have 23 pairs. So this XY right here makes the 23rd pair. That's a male because it's got an X and a Y. So this is a picture of a human chromosomes. It's called a karyotype. So highlight or underline somewhere right in. There are 23 pairs 23 pairs of chromosomes. And so this is where you can see how the DNA coils up into a tightly coiled chromosome. That happens during prophase. Oh, there it is. I knew it was somewhere. Let's see. 23 pairs of chromosomes. So if you have 23 pairs of shoots, that's a diploid. In sex cells, there's only one pair. So they have 22 total chromosomes. Where diploid cells, maybe write in the word diploid. Diploid cells have 23 pairs. The sex cells are what we call, the opposite of diploid are called haploid. And they have 23 total. The last one makes up the sex. So you can see there's 22, and then there's X and Y make up 23. When we talk about cancer cells divide rapidly, and they don't have those checkpoints that we talked about at the beginning. They don't start and stop. Cancer is this disorder where your cells are mutated, and those mutated cells keep dividing. Benign, know these vocabulary words. Let's see. Highlight divide uncontrollably. Benign is an area of abnormal cell growth that usually does not grow larger. Malignant is when the abnormal cells fail to respond to inhibitors and they keep growing and growing. Malignant comes from a Greek word like a crab. It's crawling and spreading. And so this is a picture right here of a malignant tumor. This is a picture right here of a benign tumor. I don't see where it says that, but it should. I was reading the words. So there's the new cell, and you can see the cancer is spreading in that last one. So normal cells have these checkpoints. Normal cells and tissue growth depend on balance between checkpoints or signals, and cancer cells usually don't have those. So we have proto-oncogenes. ONCO is cancer, and they prevent the cancer. They have checkpoints, and they code for proteins that promote cell cycle and prevent apoptosis. And so apoptosis, remember, was programmed cell death. So this can become overactive, and they can become cancer genes turned on like a gas pedal. Just go, 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 go. Rapidly divide, but hopefully we have the genes that stop that. We have the genes called tumor suppressor genes. They inhibit. We talk about mitosis is eukaryotic cells, because like the nucleus disappeared, the nuclear membrane disappeared. But this is bacteria or prokaryotes do something different. Highlight, highlight, here we go. They do binary fission. Bacteria don't do mitosis. They do the same thing, but it's more simple. Without all the organelles, bacteria don't have organelles, remember. Bacteria do binary fission. It's still a diploid cell. It's still asexual, means one cell becomes two cells. They don't need a partner. The cell size doubles. It reproduces two identical cells. And if you take micro, you can learn about different ways that bacteria do change up their DNA, but all bacteria do binary fission to reproduce. Okay, let's review really quickly about mitosis. Is they produce two identical diploid cells, copies of DNA. They are diploid. Promotes growth, maintenance, and repair. The next chapter we're going to learn about is meiosis, and it's going to be your sex cells, haploid and sex cells, and they produce egg and sperm. So that's the next chapter we're going to look at. Mitosis is diploid cells. They promote growth, maintenance, and repair. Meiosis is haploid cells, and it is sperm and egg. I hope this was helpful.